All right, so I'm just sort of walking around in front, and I notice that everybody's eyes and sometimes people's heads are following me. So the point is that everything about sensation is active. Your eyes are following, okay? We have a corgi, we had a corgi, Ava, and she'd hear me get up in the morning, and her ears would be, and she'd sort of track me, and if I got close to the kitchen, she knew that the food was on the way. Okay, our heads rotate. We caress things to touch them. If we were rodents, we would be whisking whiskers back and forth. If we were flies, we would be driving our halteres back and forth. Lots of animals, virtually every insect, crustacea, have antennae which are constantly moving and scanning the environment. So the point is that motor control, motor actions, okay, are an essential part of sensation. And this raises a lot of computational issues. I mean, one is, what might be the optimal way to move the sensors? Now, it was fashionable for physicists getting into biology to worry about optimizing things. I'm under the impression that if there's a bear running after you, you just basically want to look at the person next to you and be a little bit faster. Okay, so I think biology takes engineering solutions, not optimal solutions. But you do want to know something about the physical environment, about the biologically augmented environment, what the statistics are of that environment, to know how you should sense it, and then how you should use your motor actions to sense that environment. Okay, the other thing is that, so this is a topic of our program, and so I'll talk about some of these issues. And another aspect of self-motion is this idea of combining sensory channels, okay? So, you know, nature just does stuff, right? There's a lightning storm. So all of a sudden there's a crash, there's a loud sound, there's brightness, the wind picks up, you're hit with this wave of heat, the ground even may shake, right? And all these are coming into separate channels, but they're part of one process. And at the end of the day, you have to say, lightning, okay? So you have to pull this information together. So it means it has to be, so your sensors in your body are sensitive to different wavelengths. Some are sensitive to physical motion, some are sensitive to radiation. But at the end of the day, you have to pull these channels together. So there's a coordination of all this sensory information, which again is part of this process of active sensing. Okay, so another aspect of this we have to keep in mind is if sensors are moving, how can I differentiate what's self-generated movement from something that comes from the outside world, right? So there are algorithmic issues that have to be solved, either by having separate sensors to disambiguate this information, okay, or having some other clues. So I'll try to give some examples. So, and the last thing that goes into our sort of analysis of active sensing is memory. So this will come up in terms of foraging, in terms of navigating, okay? And I mean, this is an old topic in biology. I mean, the most famous story are Berg's studies of bacteria sensing a gradient of sugar. You make a measurement, you remember a concentration, like the number of receptors that are activated. You swim, some Poisson clock goes off, you make another measurement, and you decide bigger, smaller, right? So you kind of remember a couple of bits, and then you make a comparison. If you're trying to navigate in the world, you could even either have algorithms, like reflexive algorithms that don't have any memory, or you could have memory of what your previous path was. But memory has a price, okay? So just sort of keep in mind, okay, that we're really after understanding this motor control of sensors to act as efficiently as we can in the world. Okay, so the first thing we have to understand about the world is the statistics of the world. Okay, so I will blame my colleague Tanya Sharpie for making me think about this, but let's try to understand this. Okay, so another fashionable thing that happened maybe 2 decades ago in biology is people would go into the forest and take snapshots like crazy of natural scenes, or they would have a tape recorder and just tape all sorts of natural sounds, tape natural sounds in the city. I mean, talk about a great NSF grant to get. Buy yourself a Nikon camera and take Kodachrome images throughout the forest, okay? But what you find, okay, if you look at the power spectral density as a function of, say, wave number, okay, or obviously log, is that these things fall off something like 1 over k squared, or something close to this. Now, what does this tell you? Okay, I mean, it tells you if you try to find the mean, right, of this number, and the spectrum is falling off like 1 over k squared, okay, dk, that this thing is going like the log of k, which means it's diverging, which means the mean is not even defined, and certainly the variance is not defined. So maybe it falls off a little faster and the mean is defined, but for sure, okay, whatever it is, the variance of this thing is clearly diverging. So on the one hand, it's telling us that the natural world is very rich. Okay, this applies to sound, just replace k with f. The natural world is very rich. It's not like throwing a rock and having one k vector go out, okay? But it's also telling us that we're dealing in a world where even the variance is not even explained, okay? So let's, I thought I'd give an example, and sort of a pretty example, since the underlying physics is understood. And actually, this is something that was studied by Sang-Soo Kim, who's here at UCSD. And this has to be, we haven't gotten to biology like odorants affecting the world yet, but just the physical world, like how would you navigate towards a target and, in a sense, be insensitive to new stimuli. So you're here, okay, you, in this case, is going to, I'm not allowed to write up there?
You can write it.
Oh, I can.
Just don't try to pull it down.
Okay, well, no. Okay, so this is a fly flying high. Once it get here, right? So there's some heading vector, okay, and then there's the fly is going off in some direction. So the fly has this landmark, okay, and it wants to get there. But the problem is it also wants to get there in a way that doesn't really care about the details, okay? So you want to actually be able to find a source in which the details don't matter. Okay, so again, when always hears about making optimal filters to extract as much information as possible about a target, here you just care about the direction. So you want to throw away information, okay? And also, if something goes blank, all of a sudden a cloud comes over, you lose sight of the target, you want to have a system which has a built-in memory to get to that target. So let me just sort of sketch roughly then how the fly could do this. All right, so I'm going to go back here. Okay, so within the fly, okay, this really has to be one of God's gifts to experimental physiologists, okay? I mean, you can look at that thing, you want to draw kind of like a circle or a compass, as one might say, and you go to this thing called the ellipsoid body. And indeed, they're actually the axons of many cells. But there are all these cells, and they're all arranged. And if you've taken a good look at a fly recently, you see it looks like, well, it's a bug, so it has a bug eye, right? And it has all these omnipedia, and they span an amazing distance. They span something like 270 degrees. Okay, some of the best basketball players will actually go as far as 170 degrees. So keep this in mind if you play a fly in basketball one day. Okay, and there was this very impressive observation that if you look at this region, I'm not going to go into any technical details, but the genetics of virtually every cell has been well defined, and this is like a signature. And if you have the genetics, then you can put a tag on those cells, and then you could express a molecule that changes its optical properties if it happens to be putting out, if it's active. Okay, so so just 1 minute of neuroscience for our plasma friends. Okay, cells talk through spikes. Okay, so that means if you look at the voltage of a cell, here's a cell, and there's a voltage difference between the inside and the outside. The voltage difference is caused by a difference in concentration of ions. It's sort of like digital logic. There's higher sodium on the inside than the outside. Okay, the log of that ratio sets one voltage. There's higher potassium on the inside than the outside. They're both positively charged, so that is in the opposite direction. So that puts a negative voltage, and in some sense it's flipping between these two voltages. Not quite all the way up, not quite all the way down. And then there's an instability that takes place so that, okay, so there's a threshold. Here's this so-called potassium reversal potential, the sodium ion potential. There's a peak here, and we just call this the peak of the action potential. You'll see in a second. Normally, voltages so this would be a time axis, say. The neuron has many inputs. These are coming in as currents. They're charging up or discharging this outer membrane. Okay, and these values are fluctuating around. And so this is time. Things are fluctuating around. At some point, one of these fluctuations may cross this threshold level, and it crosses the threshold level. It causes this instability, which people call a spike, or an action potential. And what this does is this jumps to up a level which is about 2 ke over E. So the point is that it's a big gap compared to sort of thermal noise, and you get a spike. And then the spike is just high enough to cause synaptic transmission to occur. Okay, so this is sort of the basis of communication in the cell. Now, in a perfect world, you could record a change in voltage, but in everything we're going to talk about, every time there's a spike in time, there is a ion channel called a voltage-gated calcium channel. And you get this little bit of calcium that comes in. It's kind of like a ghost of the action potential, but it's filling the entire cytoplasm. So instead of making an optical measurement about just a layer, you make it throughout the volume of the cell, and you measure calcium, and you declare there was electrical activity. Okay, you have about a 50% chance of being right, but that's where neuroscience is right now. Is that an agreement? Okay. So, long-winded discussion, but basically then you find that there's an active region of calcium which you interpret as the activation of neurons in this body, and as you move around this body, the angle, okay, that angle theta is being encoded, okay, relative to this heading. Okay. So this is now, nature sort of solved the problem, and what's interesting about this experimentally is twofold. I mean, one is that it's pretty invariant. These are experiments that were done in the laboratory. They're usually done with stripes as opposed to a mountain, but thin stripes, thick stripes, bright stripes, dim stripes, you get the same result. So you have an invariant. The other thing is if you put a distractor, okay, you know, we saw this great film before, Formula One racing in Claire's talk. And if you noticed, okay, there are all these side rails that were put up, but it was all the street markings from the original streets were there. And if you looked at the street markings, you would have killed yourself, right? If you tried to follow any of the lines, you were just looking at the edges. So those are distractors. And somehow the human visual system completely ignored the distractors, or at least in the part of the clip that we saw, okay? So the thing that happens here is we could put on a weak distractor, and there's still one bump of activity. Okay, so this is nature solving this problem, and also, again, if a cloud comes over, and the bump will still persist. So to understand this in a kind of theoretical way, there was an idea, okay, and I'm just going to sketch it, for how you could have interactions between these neurons. So this came from physics work by Chaim Sampolinsky, who's run workshops here, and it seems to match. What's also very amazing is a couple of months ago, the complete wiring diagram of about half the fly came out, or maybe say the incomplete wiring diagram of the fly, but one half of the fly. And now there's a full wiring diagram that just got put on the archives. And so what's borne out is that you have a picture, looks something like you're used to from Turing. So this is one neuron, like represented by one fuzzball in the lab. So this guy makes bidirectional excitatory connections with neighbors. So let's call this guy some theta, and these are all its neighbors. So there's some region of excitatory connections. So these are connection strength. Okay. And then this guy makes inhibitory connections, etc., with other neighbors, like so. Okay. And if you're a physicist, what is this? I mean, the first thing you do is you take the Fourier transform and you keep the lowest component, and everything is a cosine, right? You need some kind of symmetry because it doesn't matter whether you veer to the right or veer to the left. So in this kind of picture, okay, this was 1/4 course in 2 minutes, okay? You basically have two ingredients. You have an interaction that maybe looks something like a constant, and then what you're doing is I'm getting input from all my neighbors, so I'm going to have some term that looks like my angle minus my neighbor's angle, d theta, maybe scale it, okay? And then I'm going to have some input that again maybe looks something like a constant plus something that looks like, let's just say cosine to make life easier, of my angle relative to the heading, okay? So the total input looks like these two terms, okay? This would be the external. Okay. Now, let's see where we want to go with this, okay? Where we'd like to go with this is to say, how could these interactions give me something that looks like what's seen in an experiment? So I could stretch this out, if you will, and I have some bump of activity as seen in an experiment, and I want to see if theoretically I can enforce really having 0, okay? So you could imagine, how do you solve this? This becomes a typical sort of mean field approximation, I'm just going to rewrite this total input is going to be some constant times something like the first moment, which I'm just summing up the outputs. So let me just, okay, I'm just summing up. This is the sum over all outputs, or the 0th order parameter, plus something else, which is going to look like a sum like this times, you know, something that sort of breaks the first Fourier component, okay? And so then I could, this would be R1, this would be R0, this has to have our, we end up with an equation that looks like this, and then I know I need a 0, and what I erased before is I told you when the input is below a certain number, the neuron is off, okay? So I could I can write something here that I could say my, the output, the rate of spiking of a given cell just looks like something like this. And let's just call it a threshold, where if I go back to that model of a neuron, it's off as a function of the total input, and then maybe it's supposed to fire again. Okay, so now, I mean, you could kind of get the idea that I have a rate, I write it in terms of order parameters, and now let me just scoot back here. Just, you sort of get the idea then, I could just write something self-consistent, okay? And just writing something self-consistent is just doing this sum over all my outputs, which themselves contain R0 and R1, okay, plus any input, external input, and I just threshold this. These guys appear on both sides. Okay, so this problem was solved, and there's a few things that are impressive. I mean, the first thing is it gives you all the flavor of memory that you see in the experiment. I think it's nature's simplest navigation scheme, okay? I mean, of course, the real system is not quite cosine, but that doesn't really matter. The other thing that's very important, which was an experimental test of this kind of memory system for navigating towards a goal, very active, is that suppose all of a sudden I twist the fly, okay? So now it's moving like that, okay? So that would have...
A short question, so then just to not get lost. So just, you are talking about a model of an ellipsoid body that is like a part of a fly's brain.
Yes, a part of a fly's brain,
So,
yeah.
and it really looks like an ellipsoid.
Worse, a circle.
Circle. Okay.
I tell you, the first time I saw, personally, just to interject something, the first time I saw this, it was like amazing. There's no reason in the world you could have done the same calculation the way graduate students in my lab build their electronics. They lay them out and they take all the wires and they smash them so they go into one of these little mini boxes. Okay, so then you would have had to decipher everything. But it didn't work that way. They really splayed out like this in an orderly fashion. Yeah. Okay. Okay, so in a picture like this, what that means is that you're all of a sudden trying to go from this target to that target. Okay, and in these kind of models, interconnected models, okay, rather than one peak going down of activity and another peak going up, okay, the prediction is, even though all of a sudden you move the fly, or equivalently just jump the landmark, either one, okay? You would expect within the context of the models, you go through all these intermediate states. And that, in fact, is what was seen in the laboratory. So I would say this is, just to give you a flavor for dealing with this question of how we navigate in the physical environment towards a target, we have experiments, physiological experiments. We have a wiring diagram, thanks to Davi Bach, Sebastian Sung, that is consistent with this simple picture. And we have a kind of, stat-mech kind of view of how this system works. Whoa, time goes fast. Okay, all these notes. Okay, so the next thing I just want to bring up here is how do we deal now with the bioenhanced environment? All right, so give me a moment to get a little bit of board space.
This seems very different from the statistics
Sorry?
It seems very different from the statistics you mentioned at the beginning where you had, power and height.
Okay, I'll get to this in a second. Yeah. Okay, so the bioenhanced environment. So we're seeking a source, or a predator is seeking prey, or seeking a mate. So I think the ICUP committee would have trouble if we were doing experiments with carnivores in a laboratory. Quite the bloodshed. So we tend to do much more modest things in the laboratory. And a modest thing might be this. You have a source of odorant. I mean, it's always portrayed as like a perfume or an attractant, a pheromone. So it's at first the odor's coming off. The trail is constant, okay? So you're sort of in a near field kind of view, and I mean more so if the animal can just see it, right? It's going to head towards it, okay? And then it starts to break up. I don't know, sort of drawing little wispy pictures as to what's happening. And okay, so to get back to your point, I'll say this is kind of this intermittent range. And what that means is, I mean, there are, as Massimo explained, put it nicely last week, there are regions in space where there's exactly 0 molecules of this odorant. So what could happen is if the animal is flying, it's no longer doing sort of gradient sensing, which it could if it's very close, but it's trying to get some information as to where the source is. Okay, so something, we need to add one measure. I mean, clearly if there's a plume, right, there's a wind. Okay? So first let's just look at this situation. So this is the case where, again, if we plot the log of the probability distribution function versus k, we're kind of in this range, which is people call the inertial range. And I mean, again, the variance is completely ill-defined. Okay, the mean is ill-defined in this range. So there's sort of the bigger problem here is as the animal tries to search, okay, it could search in a way that's kind of algorithmic without memory. That means it sees an odorant, it can turn in one direction or another. So you would call this like a reflexive way. Or the other possibility, it could actually keep some memory, okay, that there was a plume or a little bit of odor from that plume, which means that when it leaves that odorant, okay, it knows how to maintain its heading, okay? So you want to ask if there's some memory. And the picture that we've had, and this goes back to work by Massimo and Boris on infotaxis is, sort of framing the extreme case of using an algorithm to optimally find a path back versus, again, I mentioned reflexive, which has a much lower load, much lower computational load and much lower memory load. So the picture that one has is is this idea, you're moving along, you hit an odorant, you fall out of the odorant, and then you begin to do what's called casting, which means that you go back and forth. Just think of this as applying to many agencies for your grant, okay? And you will sort of cast, you'll hit an odorant, and then if the odorant was like this, you may get out of the odorant and you'll fly straight for a while. All of a sudden, there's no more sensory, no more olfactory information comes in, and you'll start to cast, and then finally, when you get close, you just do gradient tracking or just vision. So there's, just to put this picture together with the previous picture, so I talked before about this ellipsoid body. Okay, so what is the evidence for memory in this system? So in fact, when you cross a plume, very nicely, Emino showed that flies will actually sense smoke, seems valuable around here, which allows you to track the plume as well as the fly, okay? Then they'll actually keep a memory for a while. And so if we go back to the fly, okay, I'm going to draw my best fly. Okay, this is called the procerebral, forget about the names. Okay, there's a region here which is called looks like a fan-shaped body. I was very pleased to learn that the anatomical names in the fly are as bad as the names in the brainstem of mammals. Okay. Now, this is kind of interesting because what's happening is that this is flight information. Okay? At this point, I'm not going to discuss it, but wind information comes in, right? The animal has to correct. What? Okay. Okay. These are groups of neurons. Okay. So this place is where the outputs of neurons, okay, come together. So there are neuronal bodies doing the integration I described before. Okay, they're getting outputs from the procerebral bridge. They're also getting outputs that have to do with olfactory information. Okay, and they're combining this information, and they have this sort of regular topology of making pairs of cells, which I'll describe in a second. So, I mean, in the mammalian world, people call these nuclei, just clusters of neurons that have an identified sort of geometric pattern. But it's basically neurons. So neurons just tend not to be randomly distributed. They tend to be distributed in packets. You know, in a mammalian system, often packets are discriminated because the shapes are slightly different. The pattern of wires coming in are different. Okay, I think in the fly, it's particularly well described. Just that what you look at the distribution and you're sort of nodding your head, you're not happy. Okay. Can't satisfy everybody. Okay. Okay, the point I wanted to make.
So, ask a question. So it seems that the fly has a special part of the brain responsible only for navigation, right?
It has specialized parts for everything.
For everything.
Yeah.
What's about mammals? Do we have like ellipsoid bodies?
Well, there may be something like that for heading, but at least as we see it, it's not as aesthetically arranged.
Okay.
Okay? So nothing that we know about in mammals is it's more amorphous. But like I said, it could have the same function. It'd be perfectly amorphous.
So it is a specialized device. So it's not like a CPU, which analyzes all the information. It's like an autopilot. So just a separate part.
The CPU is a separate part.
Yeah, we could argue, and Marcus is here, we could really argue whether there's the equivalent of a CPU. But, okay, I'm just trying to give a sketch. All right, let me go on for a minute before we run out of time. Okay, so I described a system before, which is capable of memory, and it was a distributed system. So what is the fly doing in order to keep some track of the confluence of wind direction and olfaction? So details aside, it's getting wind information, which is coming in like this, and it's getting olfactory information, which is coming in like this. And then if we were to look carefully at these cells, interestingly, these guys form a latch. Now, the usual kind of latch we're used to is inhibition, and that's a flip-flop, okay? So we're just saying that the output of one cell is, again, maybe some nonlinear function of some connection strength times the output of its neighbor relative to a threshold. And this guy is the same thing, but it gets input from the other neighbor minus a threshold. And this guy has outputs, two stable states, 0, 0, 1, 1. So if the wind is blowing and olfactory information comes in, these guys will be active, okay? And then if you lose the olfactory signal, it's still stable. And then sometimes later, the signal is gone. So this is a second example of a memory system that is, plays an essential role in navigating that. So rather than, in this case, heading towards a fixed object, which is not moving, I mean, for which there's really no statistics per se, except for clouds or whatever, here, this is navigating in a very complicated environment, okay, and having a minimal of memory. So this is an open question. How do you trade off your search algorithm and the price of building this whole structure to do memory? Okay. How religious are we with time?
On when you said it's still stable, like you pointed an image basically this sense where I guess it still has the value 1.
Yeah.
The factory information changed, but then something about what the.
So if the wind is still blowing, okay, so this guy, well, if you would say the cell is off, and then it's on here, it's the excitatory input to that cell has dropped, so it's dropped to this point, but it's still getting excitatory input from its neighbor that keeps it at this point. Then if the wind was to drop off, the input decays away and it turns off.
So
Okay.
both inputs have to turn off before the cell
Right.
is active.
Okay, there's actually an inhibitory reset, but it boils down to both inputs turning off. Yeah.
Okay.
Okay. I'm being a little, not glib, but just a little bit fast.
What is the characteristic time at which the information is renewed? So just for how long? So just will this neuron retain this information? And
Oh, someone has to help me on that. I thought it's on the order of sort of 10 seconds, right? 10 seconds would be about a centimeter or so, a flight. Yeah,
10 seconds. Okay.
yeah.
So and in normal,
But the neurons, just keep in mind, the neurons themselves are kind of like 100 millisecond devices. So you're picking up 2 orders of magnitude by this memory system.
okay.
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 Okay. So, okay, the other thing that came up and as an issue is what happens when the wind dies? What happens when the wind dies? So these pictures are kind of looking down, from the top, right? But if we look at, sort of Z, okay, what's happening is that the fly is doing its thing, but the fly will start to drop. So we need to understand something else about the plume. And this sort of begins to bear on the kind of statistics used by mammals. So if the plume in fact, gets close to the ground, okay, it gets stuck. So plumes that touch the ground can leave molecules on the ground. They're kind of frozen, okay, so they're a weaker concentration, a lower concentration, but they're not fluctuating. And so phenomenologically, it's interesting that when the flies will head towards the ground when they lose the wind, okay? So you pay a price if you search along the ground. It's very local, okay? But it's relatively stable. Now you see the same behavior actually in rodents and apparently in dogs, okay? Because if the trail is strong, it's nose to the ground. And if the trail starts to break up a little bit, they'll actually start to cast, okay? But if in fact the trail disappears, they'll rear up, okay? And they'll start moving their head from side to side. They've lost the trail, they need to span, need to be sensitive to a much more global region in space. So that was, this is work, I would say, in progress. You might say this is so, to understand what is relative height off the ground, okay, in terms of navigation. So this is sort of open issue, both in terms of experiment and in terms of theory. Okay, how am I doing time-wise? Okay, all right.
When you say it's an open issue in the context of your field, do you mean with respect to the diagrams that you've drawn about basically like what the neurons actually do, like what sort of like signals are being sent for how we do activation, or is there something else that I missed about basically what that means that it's an open question in y'all's field?
I think what it means is that we only have very few studies. Actually, both people who did these studies are in this room. You know, so there's 2 physics retreads that have worked on this problem. So we'd like to understand exactly in typical environmental situations, if you could say that, okay, how you deposit clues onto the ground, and then experimentally what the strategies are. So we could get some better clues to constrain theory. Okay, so that's what I mean by an open issue. I think you're also asking, does this mean we need to learn something more about how the nose works or something like that? So I'm not, yeah, I think, no, okay. We need to understand more about the statistics of search. All right. So, I mean, the good news and the embarrassing news that it's only been 20 years since people realized that doing experiments on anesthetized animals is sort of like studying a toaster with the plug pulled out of the wall. So we're starting to make progress. Okay. All right. Let me. The last thing I just wanted to touch on is, so I've sort of talked a little about memory. I want to talk a little bit about coming to grips with self-motion. And again, I'll try to pick 2 examples, one from the fly and one from the rodents. So give me a moment here. Okay, so birds fly with 2 wings. How do they get away with this? Okay, what was the critical thing that the Wright brothers actually historically noticed about bird wings that sort of eluded everybody else? Control, right? They change, okay? They actually change the shape, right? They change airflow over their surfaces. They're continually warping their surfaces, right? Keeps them from stalling, et cetera, et cetera. So flies, at the first approximation, do not change their shape. So they need an additional gizmo, okay, in order to get feedback, because they can control an angle, right, and they control the depth of their flight, but they can't control the shape. And this brings up a wonderful gizmo called the haltere. Okay, and I'm going to contrast this in a moment with something called the whisker, which you heard about this morning from David Golem in rodents. Okay, so this, the fly wants a gizmo that basically controls its heading. It's sort of the vestibular system, if you will, of the fly. And I think the one lesson, at least that I got out of this meeting, is like the most, at least as a mammal, the most important thing is your vestibular system. Because otherwise we'd be crawling around on the ground here. And we wouldn't be worried about the statistics. You'd all be on the ground. Okay. So you need something that's for the fly to know if it's rotating, and that's independent of any landmarks. That could do very fast control. And this seems like a problem right out of Physics 1, right? Textbook. So just set up a little coordinate system here. Okay, so what we're going to do is we're going to literally take a ball at the end of a stick, and the ball at the end of the stick is going back and forth. Okay, so it's rotating. It's rotating along z. So there's some angle momentum here. So this guy has some angle, maybe, which is going like sine omega t. So this guy is having some angle momentum, which looks like that's my omega. Okay, and this is pointing in the x direction. Okay, so far trivial, right? So the question we're asking is, what happens now if the fly is trying to turn? So now the fly is trying to turn, right? So it's trying to turn with some omega. So we have to calculate the torque. Okay. Well, you just get this, right? Omega, omega. And plus, we get this extra term here, right? Which is just omega cross L, and this would be mr squared, little omega, big omega. So which direction is this? This is in the y direction, and this is cosine omega t. So it's basically, let me just move one omega. It's basically these two terms. So this is very nice. The animal, on the one hand, has a self-reference term. Okay, so it has force transducers. Okay, it has one self-reference term, and then it has a second term that depends only on its rotation speed. Okay, so this, in principle, I mean, this remains to be seen experimentally, this could serve as a reference term, okay? And then this term tells the animal that it's actually rotating along this axis. So this is a simple example, I'd say, of how an animal uses something that looks like chain maul from the Middle Ages to correct its direction. These are fast. These are about 250 hertz, and this guy is a comparable change in angle. Okay, the vibrissa in the rodent has an interesting problem. It's tapered, okay? And here it's also, you might say, rotating into the board. And every so often it wants to whack into an object. And we want to know what, where that object is in space. Okay, so if it hits this object, it could become tapered, okay? And there's some sort of radius of curvature, r, and that generates a torque, right? That'll generate a torque that looks like Young's modulus times its, this guy is, it's called this z. This guy is just z squared times the area of the vibrissa over this radius of curvature, okay? But how does it know where it is in space? Okay, so in fact, as it moves back and forth, this guy has a little bit of moment of inertia, and that will also drive receptors. Now, it seemed here that nature kind of was a little bit clever because this is a cone, so its moment of inertia is much less. If this was mr squared and the total mass here is m, here the moment of inertia turns out to be an order of magnitude less. So it's just tuned enough that you get a spike, at least in some cells, every time it goes back and forth. And then you gain these additional spikes due to this torque every time it whacks into an object. And that's at least one example where it gets back to how you could utilize actually self-motion. Okay, so I gave one is with the haltier, that you get a calibration signal. It's orthogonal to the direction of rotation to the animal. It seems in this case, maybe the vibrissa is not as clever as the haltier, but one's a high frequency signal, the whacking into an object. One's a low frequency signal moving back and forth. And this provides a calibration of where the whisker is in space. So these are some examples. All right, so maybe to get back, this is looking at animals in the natural environment, the ability to track them, the ability to record from the neurons involved in different circuits is, I don't want to say it's crude, that's sort of insulting all of us, but it's really, we're really at a very primitive state. Okay, I mean, the fact that the first true wiring diagram of the whole animal is only happening now, okay, and that the wiring diagram of the rodents, say, looks like it's a decade ahead of us, it just sort of puts this in perspective. But I think we're running a workshop, we're very optimistic that we are sort of forcing the field in a way to deal with how animals interact with their natural environment. And we'll see how this impacts the rest of the world. Okay.
Could you just re-summarize the durability of memory in different types of animals and for example, visual memory versus other types of sensory memory
I mean, just like a little pedagogical tour.
The fly is very short-term memory compared with even fish.
Well, I actually, maybe somebody else here knows, I don't know what the effect, I don't have any knowledge of long-term memory in the fly. Or even if people have done LTP experiments in the fish. I mean, synaptic physiology is pretty much the same in all animals. Okay, so there's two ways. So the examples that I showed were very much in this realm of what people might call working memory. It depends on the electrical activity of the memory. If you turned off the fly and then you turn the fly back on again, gone. Okay, and there's evidence for this kind of short-term memory, acting on kind of tens of seconds scale at most in you and I. Right? You could do the simple experiment that Dave Robinson did many years ago. You fixate on an object, you close your eyes, and your eyes don't drift until about 20 seconds. So just phenomenologically. Okay, so there are circuits that look like they have this kind of recurrency that we saw in the ellipsoid body. I mean, long-term memory, the experimental evidence is that it is a matter of changing synaptic weights. And there is nice work that shows how the confluence of activity of the neuron receiving the input and the neuron giving input could lead to changes in weights. And again, I think here, neuroscience really has to go further because beyond, sort of two spikes, we don't have good statistics on how detailed spike patterns actually affect long-term memory. There's trends. It's like culottes, right? There's trends. Okay. I don't know if this helps. Yeah.
Physiologically, where is this organ located in birds? Is it a part of a brain or?
Which one? Haltier?
Yes.
No, not bird. That's a fly.
It's a fly. Okay.
Okay. So
Where is it?
it's right here.
Okay.
Okay. So next time a fly goes buzzing around and your first Well, first of all, if a fly has 4 wings, it doesn't have a haltiere. The back haltieres actually were formed as vestiges of wings. But next time a fly is buzzing around and it disturbs you, you can take a cross product or something. Before you, like that, you should look at the fly.
So, and so just concerning the whiskers, so just if you cut the whiskers off, so that the animal is lost. So just.
Oh, don't do that.
No.
I mean, that
By
should.
no means, just it's like an academic question.
Yeah, I mean, okay, so actually, if, are there any Ayakak people? No. There's actually many rows of these things, but this is supposed to be a mouse. When they walk, the bottom rows are face down. You get they're really touching the ground. I mean, they're really using their whiskers as like a sea, they're moving them so it kind of covers the space that sort of an entire porcupine set of quills would cover, and it's sort of keeping them a certain distance from the walls, from the floor. You know, it's defining their interpersonal space, one might say.
But vision isn't vision not enough?
Well, there's 2 problems, and maybe Chris will. I mean, one thing is that, you know, the binocular vision very close up is not working well, and binocular vision is certainly not working off to the side, as with you. So it's as though the visual system is all the equivalent of your peripheral visual system. So it's a kind of low resolution, but to gauge this distance, you really need binocularity, and there's no evidence for this binocularity off to the side. Yeah.
How do we know that the fly is doing all these calculations? Well, let me put it this way. How would you study the brain of fly?
Okay, so, okay, I'm just pantomiming this because I studied one fly once in setting up an undergraduate laboratory. But the way most of these experiments are done is you take the fly, right? Suppose this is the z-axis in the lab. So I mentioned all experiments are done iteratively. So you make the best guess as to what group of neurons are interesting. Okay, and today you could access those neurons based on their genetic makeup. And because you know their genetic makeup, you could transform them to express certain molecules. So suppose you transform them to express this calcium indicator. Okay, so like in this experiment I described, that was first done by Vivek Jairam at Geneva, where they made a line of flies that have that you could express a molecule in that cell. Okay, so now you need to image those cells. So you go in with microscopy to image those cells. That raises other problems because usually, even though the fly is very small, the tissue is highly scattering. So there was a nice technique that our colleague Winfried Denk of doing nonlinear excitation. So you only excite right at the focus. So now what I've done is I could excite a molecule whose optical property changes at that point. So let's say those were the cells of interest. If not, you move on. Now the fly is immobilized, and I could have, it's held on a stick, and I could have a torqueometer. So this is a high impedance device, so it measures the intended torque without actually moving. It's sort of like a voltmeter for torque. So I've turned the fly into a function, and it's reporting torque. So I have another measurement, and then in front of the fly, as is done, I might have an arena. So the arena could have stripes, which I'm moving, so that corresponds to there could be one stripe here, which corresponds to the heading, the landmark it's moving towards. So that allows me to control the environment. So like an experiment I mentioned, where you all of a sudden, now I can't move the fly because it's stuck on the stick, but where I move the mountain, which corresponds to moving the stripe. But because I control the laboratory, I could just jump, okay, that stripe. So I have control over, this is over the environment. It could be a flying fly, but it's not moving anywhere. okay, that may introduce problems, right? No wind in this case, okay? I might have to introduce a wind eventually, but you sort of get the idea.
But the fly is under high stress.
How do you know?
You're doing an experiment.
Well, I mean, maybe. I don't know. It's a fair point. So you could say that in animal experiments, like rodent experiments, you could mention we could monitor steroids, which we have in some experiments. The only experiment I know in which a rodent is really under high stress is when you put a rat or a mouse in a magnet and you do fMRI. Because in an fMRI machine, what you hear is due to the gradient coils. And if you're a rodent, you think there's a big animal on top of you that's about to chew into your burrow. But usually, the animals they become head fixed, imperfect, right? No vestibular system, okay? But they're being rewarded, and you know, we could do certain controls. And then you could ask, I mean, as like Chris did, you could try to relax the environment. We could make a less perfect measurement in an open field environment. So with an optical measurement, okay, clearly I can move anywhere in the brain that's within my field. If I stick a wire in, I'm stuck in one spot, but you know, I could verify things. So I think there's some, people worry about this. I mean, these are all very fair points, but every experiment is compromise, right?
That's a great moment to stop. Thank you.
Okay.